my dad, Dr. Ron, and so, but we're really excited to be presenting not only a lecture together, but in particular this lecture. Since we kind of came up with the idea for this topic, probably about 3 or 4 months ago, every time we start talking about, something new, something in the news, a new chronic disease or a new, study, it all relates back to this. And we always kind of circle around to this epigenetics. So we've both been really excited about this topic and really excited to present. It might seem a little bit scary sometimes when you start talking about genetics, and hopefully, we tried to provide enough information on the slides, and hopefully it doesn't get too technical. If it does, just raise your hand, ask questions. If you want us to explain something in more detail, let us know. But we wanted to provide the framework for this idea because epigenetics really sets the stage for almost every major chronic disease that we're seeing on the rise today. And it seems like such a broad blanket statement, but really when you look at the biochemistry behind what's going on and why we're so sick, it all relates back to this. So
it also relates to the basic theme of the Riordan Clinic, which is helping people learn how to take better care of themselves, which means that every little choice that you make is a kind of dialogue between you and your genetic code. And your genes, rather than being a static kind of system that just sits there and kind of you manifested a certain body when you were a little baby and it grew, it's actually a very dynamic conversation that's going on between the environment and your genetic structure. And we'll get more into that in just a little bit and how that works. But to me, it's really one of the most hopeful and exciting things that's come out of the study of the human genome. And we're going to explain kind of how our concepts of what genes do and why they're so important has changed as the study of epigenetics has moved forward.
Okay, so just to give you a little bit of framework, when you break down the word epigenetics, the prefix epi means above, and genetics is for our genes. And so when you translate the word epigenetics, it really translates to above our genes. So what we're going to be talking about today is not necessarily just the structure, the DNA structure of our genes, but more so the proteins that are involved with providing the framework for that structure. So some of the proteins, the double helical structure of DNA is wrapped around certain proteins, and depending on how tightly wound that DNA is or how loosely wound it is will affect if those genes are actually expressed. So just to clarify, we're not actually talking about changing the structure of our DNA, but more so all of the proteins and all of the structural aspects that surround our DNA.
One thing that Dr. Riordan was the founder of the Riordan Clinic, and when he was in medical school, they did a very simple but yet profound experiment with mice. We don't have this in the presentation, but it's very relevant to this. They took the mice and they gave it a perfect diet. The medical students gave their mouse a perfect diet minus one nutrient. And then they looked at what happened to that mouse. And later on, several years ago, we replicated that study, and the same things there was just subtle changes, but as time went on, more and more dysfunctions occurred to the mice that was missing just one nutrient. They had all the rest of the nutrients, but they were missing one. And there was a doctor in Kansas City that took our data, and they did a special analysis and found out that just by leaving out one nutrient, there was over 500 or 600 different genetic things that changed as that mouse attempted to adapt to the missing nutrient. Because when we don't have what we need, we try to adapt. And sometimes it's a maladaptation. So
one of the best examples of epigenetics, we've known about the idea of epigenetics really since the 1970s. So we've known about this idea for a long time, but it isn't until about the past 10 to 15 years that they've started looking at this in a different way to try and explain some of the chronic diseases. But the most classic example of epigenetics at work is when you have the sperm and the egg cell, the chromosomes of the two parents that combine together to form, new DNA. At that point, you have one cell that then is able to differentiate into, neurons, muscle cells, liver cells. So from this one cell, from this one genetic code that we have, this one DNA code, by strategically turning it on and turning it off at the right times, we're able to develop into all of the different, multicellular organism that we are. So it really, that's the most classic example of epigenetics. And so we'll now kind of move on. And epigenetics really, the question is, is this a good thing? Is this a bad thing? Epigenetics allows us, like Dr. Ron was saying, to have this constant conversation, constant dialogue between our DNA and our cells and the environment. So it's an adaptation that allows us to respond very quickly to what's going on in our environment. And, this is beneficial in the sense that we can, it gives us hope in the sense that we can positively make changes in our life today that will affect our genes, but in a more detrimental way, the poor choices that we make are being imprinted onto our DNA and getting passed on to our offspring. And so it kind of goes both ways, and we're going to kind of try and more focus on the hopeful aspects of what are the positive things that you can do, to positively affect your DNA and provide not only the best hope for yourself, but also your offspring.
Here's an example of that. Oftentimes you'll hear people say, well, my whole family had heart disease, so I'm doomed. I'm going to have heart disease now too because it's genetic. And really, I always say, okay, wait a minute. Let's talk about your family that has heart disease. What did your family eat that you're now eating? Did they smoke? Are you now smoking? Did they exercise? What's your exercise habits now? And very often, whatever you grew up with in your family, that becomes your behavior, and that becomes, in a sense, the epigenetic impact upon the genes that you inherited. Sure, you did inherit family genes, but how those genes how they manifest, how they express, is dependent upon behavioral choices that you make. Now, unfortunately, when we're growing up, we don't know any different. If our family's a big sugar-eating family, we eat sugar, and it tastes good. Or if we're a big smoking family and everyone smokes in the family, you feel like a weirdo if you don't smoke. And so a lot of genetics have to do with behaviors that have an impact upon how the genes express.
Okay, so we're going to take a step back and do a real quick brief high school biology, DNA, genetics lesson, just to kind of, so we understand, kind of the framework for epigenetics. DNA.
Nomenclature.
Nomenclature, yes. So we have, the DNA that is packaged in 23 pairs of chromosomes. Like I said, each set inherited from our parents, and we have, these nitrogen-based building blocks that provide the code. And it's really what's interesting, what's fascinating about this is it's the sequencing. We take these four nucleotides, bases, and the way that they're sequenced is what provides the instructions for our, as our DNA. And the size of a gene, what's interesting, you could have 1,000 of these bases, and that's considered one gene. You can even have up to 1,000,000 of these bases, and that's considered one gene. And so it's really an important sequencing and timing, that affects how this DNA is expressed.
It's another interesting fact is that they're talking about using gene sequencing as a way of coding books. You can take the digital sequence of these genes and you can take a book and put it in biologically and then read it off later on and get the exact same book. It's that precise. And so they're saying that there may be biological computers in the future that use this technology to store information, because think how compact so much I mean, a whole human being is stored within a microscopic cell, you know, and it's amazing. So when we go on, go ahead.
Okay, so how exactly does our DNA work? How exactly does it, because you know our DNA provides the template, but it's really through a lot of different messenger RNA type systems that this code is actually read. And so you can kind of see a picture. Our DNA is, you know, unzipped and then it's copied, and then that copy is then translated by the messenger RNA. So there's a lot of steps that go into, you know, not only correctly providing the information, but also correctly translating the information.
How many of you remember when you very first kind of somewhat understood how DNA works? And maybe now you're feeling a little bit fuzzy about it, but it's really very simple. The DNA has the coding here, and when it splits apart, and another set forms on the other side, so that you have two identical sets. But that's when we're being born, when the germ cells are being made for, you know, the mother and the father. But then there's another type of DNA called RNA that forms a template that the body uses to make proteins, and this template codes for the proper sequencing of proteins. For example, let's say insulin. I think insulin has 160 amino acids or something like that. And so when your body wants to make insulin, it takes that part of the DNA and opens it up, and then a little messenger RNA comes in and forms the template, and then it floats out into the cytoplasm of the cell and starts collecting amino acids in the right sequence, and there's various enzymes that put them together, kind of like little building blocks, little Legos type thing, and all of a sudden you've got insulin. So there's over 50,000 proteins that your body needs in order to constitute life. So this is going on in our cells all the time. And so there's the DNA, and then there's the messenger RNA. And the main thing that we thought is that DNA was only for the purpose of making these correct proteins. But there's a whole thing to the story. So this is going beyond. This is where I put down here, genes coding for protein synthesis is now considered an oversimplified view of what genetics is all about. It turns out that, you know, of these 6 billion bases that the human genome has in it, only 1.5% is used for coding these 50,000 proteins. So people begin to ask, gee, what are the rest of these genes doing? The ones that are coding for proteins they call exons. And so this part of the DNA is the exome, you know, the genome. Now this is the exome. This is the part that's coding for these proteins. So what's the rest of the human genome actually doing? Has anyone heard about this yet? You're going to learn something new today. It's really cool. So most genes are what's called regulators, and they are called intron genes or promoter genes. The promoter genes turn off and on the process. The introns do the regulation. So that means that 98% or more than 98% of our genes are actually helping to regulate the process of how the cell works, how proteins are formed, the timing of how the proteins are formed. So they're also monitoring the genetic code to make sure that if there's any damage to the DNA code, there are repair mechanisms that can go in and repair the template. So there's a whole bunch of other things going on with our genetics besides just the making of proteins. It's the regulation of proteins as well. So they're now saying that there may be over 100,000 of these RNA regulator genes. And so coding for synthesis is the old dogma. The new understanding is regulation, the regulation of what's going on. Now, why does this make sense, or what does this have to do with our conversation today that's really quite exciting? If you think about what are the major diseases that our culture is dealing with, it's the diseases of aging. Let's just take one very common disease that I deal with a lot every day, and that is fibromyalgia. How many of you have heard of fibromyalgia? About 9 out of 10 people that have fibromyalgia are women. Men get it as well, but it's a very complex illness. Most doctors don't really know very much about it. It's associated with a lot of problems. There's pain, there's gut dysfunction, there is problems with brain fog and immune dysfunction, fatigue. Anyway, there's a number of dysregulated functions within the body. So they're changing the name now to from fibromyalgia to the dysregulation syndrome. And when I heard about that, it made me start thinking, my gosh, most of the problems that people go to doctors for now, let's take hypertension. That means your blood pressure has become what? Dysregulated. Or let's take arthritis, the normal functioning of joints. It's an inflammation, right? And many times it's an excessive inflammation. I wrote a little book on inflammation. It's where the normal inflammation has become dysregulated, and now you've got too much inflammation. Or just a more common example that would be appropriate for now would be allergies. It's where you've got your normal inflammatory protection against antigens, but if you have an exaggerated response, you have an allergy. That's where your immune system has become dysregulated. So this is why this keys very closely into this concept of epigenetics, is that there must be signals occurring in the environment that is causing our normally well-regulated system to become dysregulated. This is actually the story of epigenetics. So I use the example just to kind of give a real-world analogy to these exomes and introns. Whenever we hear a new word, we go, oh my gosh. The exome and the introme, excuse me. The exome would be like the word. If you have a computer, you've got your programs stored on the hard drive. They're just sitting there. They're not doing anything until you click the button that activates the program, until you touch the app in order to activate the program. Once you activate the program, then you start putting in your specific keystrokes. And if it's a word processor and you've got a homework assignment to do, that word processor could be used for hundreds of different things. You're going to use it for your homework assignment, whatever that is. So in many ways, the exome, which are the DNA that just sits there, it's waiting for the environment, for us in the environment, to go to it to get what it needs, what we need in order to adapt to our environment. So it's like a working set of information that gets activated by what's happening in our environment. So I call that like the keyboard, the screen, and us typing into it what we need from it. So that's just an analogy to kind of help that out. So I think that's going to... So the dysregulation of the DNA is where we basically go to it, and we go to our adaptive system and we overload it. More often than not, like for example, back to the fibromyalgia, one of my theories as to why women are more prone to fibromyalgia is that modern women are called upon to do everything. They have to be a mom, they have to be a housekeeper, they have to earn money, they have to go out and get a job, they have got kids, they're trying to keep the peace in the family, you name it. Women nowadays are really very much overloaded by the demands that are being made upon them. So you could think of fibromyalgia as an epigenetic overdraft. It's kind of like where you've loaded too many lights on the Christmas tree, and pretty soon the breaker switch hits, and the lights go out, and now you're tired, you can't do anything. It may be the body's attempt also to just slow you down and kind of make you rest a little bit to kind of get caught up. But this is dysregulation, and so epigenetic disease is really a dysregulation of what would otherwise be a well-tuned system.
Well, and speaking of, the body's ability to adapt, to the stressors in the environment, how many people, whether it be through the news or through your own personal experience, know of a child that has a disease or a chronic condition that typically in the previous generation we didn't see until somebody was older? So for instance, juvenile rheumatoid arthritis, diabetes, type 2 diabetes, is on the rise in children. So type 2, they used to call adult onset diabetes. Well, they're having to rename that because they're now seeing it younger and younger, in the next generation. And so the old thinking of genetics, and especially epigenetics, was that, when the mother and father's, cells combined, to form this new life, that there was this epigenetic reprogramming, where in a sense that child's DNA was wiped clean, and all of the epigenetic tags that had been tagged, in the mother or the father were all of a sudden wiped away. Well, we're finding now more and more that it's these epigenetic tags that are getting passed on, and that is starting to explain more and more why we're seeing, these epidemic, increases in obesity, why, if the parent is overweight, the child has more of a propensity to be overweight, and part of it is lifestyle, and it goes back to that nature versus nurture question, and, is it, what the child was born with, is it their genes, or is it how they were raised? And it was always kind of flipped back to, well, it's just their environment, it's just how they're raised. But epigenetics makes that connection for us, that genetic link, because we know, DNA takes thousands of years to actually change the structure of DNA, but yet we have this question of there are very obviously some chronic conditions that are being passed on within 1 or 2 generations. And so these epigenetic tags are what kind of make that connection for us, and again, what in my mind bring a lot of hope, because as quickly as you can change your epigenetics for the negative, you can also swing and within 1 generation change it for the positive, and pass on a genetic an epigenetic profile for your child that is much better than what you received yourself. So
I in trying to put this in something that I deal with all the time, which is thyroid disorders, I have a graph up here. It looks pretty complicated, so I won't try to go through all of it. But basically, what I have found is that these 5 things right here, fasting, stress, infections, toxins, and inflammation, turn out to be very powerful dysregulators of the thyroid system. Maybe you've heard of people that they have a lot of the symptoms of low thyroid, and then they go into their doctor and they have a TSH test, and the doctor says, oh no, you're fine, your thyroid's fine, you're okay. But you say to the doctor, wait a minute, my hair's falling out, and I've got dry skin, and I'm cold, and I'm very, very tired and depressed and can't remember anything, and I looked up low thyroid and I fit all those symptoms. But he says, but your TSH is fine. Well, it turns out the TSH regulates the factory and the production of the hormone, but these regulators here have a lot to do with what happens to the thyroid hormone in terms of activating it so that the body can make good use of it. Our ancestors, if they, and when I say ancestors, I'm talking back before Dillon stores were everywhere, and drugstores and hospitals and stuff. If they were starving, which would be like fasting or like being on a diet, it made sense for their body to epigenetically downregulate this 5' deiodinase enzyme. And by downregulating, it would mean that less of this hormone, the T4 with 4 iodines, less of it would be converted to this form of T3, which is the activated form of T3. And so it would have to go somewhere. It would go this way, towards the reverse T3, where it's a different configuration. And this turns out to be a blank key, so that when we finally got the hormones to the receptors, there was more of the blank keys and less of the active keys. And for that person who was starving, their metabolism would slow down. They would survive the famine or the lack of food or whatever. And then eventually, when food came back along, it would reverse. Epigenetically, it would reverse, and their metabolism would come back up. What happens to us in our time is some of these factors that slow the system down, they never go away. If you've got mercury fillings, you've got mercury in your system all the time. If you're eating out of plastic or drinking out of plastic bottles and are exposed to plastics all the time, that doesn't go away. If you've got a chronic low-grade infection in your body, or if you're chronically inflamed because of a chronic autoimmune disease, you never get rid of these particular dysregulators, and so consequently, you're never quite getting your thyroid hormones in balance, and so you tend to have low metabolism. But that doesn't show up in the standard tests. So if you're thinking epigenetically and how dysregulation may be causing your illness, that can give you a clue on how to correct it and things that you can do to make an impact upon your environment, such as to take away those environmental dysregulators that are keeping you unwell.
Again, it's this dance between our environment and our body, and in so many ways, it's an amazing adaptation that we have, that we can respond so quickly to what's going on around us. But unfortunately, with the type of environment and world we live in now, we are overloading our epigenetics, and our body is trying to compensate in the best ways that it knows how, but unfortunately, it's too much.
Take diabetes. It'd be another example of an epigenetic disease. Our ancestors, the genes that we have, we were never ever exposed to the amount of sugar, well, stress, but to the amount of sugar that currently is in the modern environment. And so when the cells are exposed to more and more and more sugar, they develop insulin resistance. And so then, as a kind of a consequence, the body has to make more insulin. It will send out a program to make more insulin because that you have to do something with the sugar. And so, but that insulin, unfortunately, increases fat storage, especially in the visceral fat, around the organs. And so we all start developing these little pot bellies, and that's an epigenetic maladaptation. We're trying to adapt to the high sugar environment, or we're trying to adapt to we don't realize that wheat, for instance, has changed, and it now has a glycemic index higher than sugar. And so here we are eating our whole wheat bread, trying to be healthy, and little do we know that 2 slices of whole wheat bread has a greater impact upon our blood sugar than a can of pop. And so there's a lot of so this is why we have to kind of use this new information to investigate our behaviors, our habits, to see if what we're doing is working for us, or if it's making our maladaptation, our dysregulation, worse instead of better.
Well, does anybody know why we like sugar so much? Does anybody know why we crave sugar? I mean, have you stopped to think about, why does sugar taste good to us? Well, yeast, yes, it does feed the yeast. But from a evolutionary perspective, our brain runs on glucose. So from an evolutionary perspective, when sugar wasn't in high quantities and easily accessible, it was an evolutionary adaptation, a genetic adaptation that we would, when exposed to sugar, want to crave it, want to eat it, because it feeds our brain. And so, but we've now our culture has shifted so quickly, and the availability of foods has shifted so quickly that our genes haven't caught up. And so, just like Dr. Ron described, epigenetically, we're trying to accommodate, for the high amounts of sugar, and our body's trying to accommodate for that being in high demand, but genetically, we haven't quite caught up to that yet. So, so kind of shifting gears, again, back to more kind of left brain, what exactly is epigenetics? And so, I explained a little bit about how, tightly or loosely wound, the DNA is. And so DNA is actually wrapped around, now let me just move on to the picture because this will show it better. If you look at,
This button right there.
okay. If you look at right here, these are nucleosomes right here, these little balls that you see, and wrapped around it is the DNA. So this is the largest view of the DNA in the chromosomal form, and then when you go microscopically, a couple layers deeper, you can see how the DNA is wrapped around these nucleosomes. And nucleosomes are made up of histone proteins. And these nucleosomes and histones are highly susceptible to environmental changes. And it's, in fact, the shape of these histones that either more tightly coil the DNA, which shuts down its ability to, replicate and express itself, or it more loosely coils the DNA, which allows that DNA to be expressed. And then even a little bit further along, you have these little methyl tags that are on the DNA too. And I'll show you here in just a second how these methyl tags affect the expression of DNA too. So when we're actually talking about epigenetic factors, we're talking about these proteins here at the cellular level and how they actually affect how well or the timing of when that DNA is expressed.
Structure and function.
Structure and function, exactly. So DNA methylation, the more highly methylated areas of DNA tend to be more transcriptionally active. So the more methylation you have, the more active. The less methylation you have, the less active that DNA.
And where that ties into nutrition is maybe you've heard how important B complex is. Well, B complex, especially several of the B vitamins like B6, folic acid, B12, these are very important methylating molecules. And so if you want to have an impact upon preserving the health of your DNA, make sure you're getting adequate amounts of B6. And just to bring this even more specifically, what nutrient is missing in babies that have spina bifida? Folic acid. That's a B complex B vitamin that's very important with methylation. And so if you lack folic acid, you can't properly methylate your DNA, and so you create the problem for a major mistake in the growth of this particular child, and it'll end up as a birth defect.
So just to give you a few kind of what I thought were interesting experiments of epigenetics at work, if you look at these two mice that are pictured here, which of these two mice looks healthier to you? Is there one that looks... So pretty glaringly, exactly. So it's pretty glaringly obvious. So they did a study specifically isolating what's known as the agouti gene. And this gene is actually present in both mice and humans. I don't think they've actually done a human study with this yet. But this gene is present in both mice and humans. And unmethylated, so basically without those methyl tags, this agouti gene is what predisposes people to obesity, to cancer, diabetes. So you want this gene to be more unmethylated. So I'm sorry, no, you want it to be methylated. You want it to be more transcriptionally active. So what they did in this experiment is they have the two mice. They first fed them both with an additive of BPA, which BPA has gotten more press lately, probably in part due to this study. But how many people have seen on water bottles now, they all say BPA free? So BPA is a component in plastics, and they've now shown that it's harmful, to us and our genes. So both mice were fed a diet with the additive of BPA in it. However, the only difference was this mouse here on the right was also fed a methyl-rich diet of B vitamins. So they also added in these B vitamins, and you can see the difference between the two mice. And what's not discussed here is even looking at the mice's offspring, that the mouse that the overweight, the yellow-coated, unhealthy-looking mouse, even though this mouse's mother was the one that was exposed to the BPA and that lacked the B vitamins. This mouse was fed a normal diet with B vitamins, and the offspring, it helped reverse the trend. And so so all that to say, you can reverse, some of the effects of, a poor diet choice in your mother.
So that's really the whole idea of epigenetics, is that we can learn about things that we can eat, supplements we can take, getting adequate sleep. Exercise is an epigenetic behavior. Certainly lacking exercise sets you up for more problems, whereas exercise helps protect you against a number of problems. So B vitamins are methylators, so that's something that you can do, and we've oftentimes referred to the B complex as the stress complex. So if you're under a lot of stress, it's when people are under stress that very often diseases or conditions manifest. And so B complex, turns out it's a good antidote against stress because it's a good source of methylation.
Mm-hmm. So again, this is another great example. You know, mice were fed a common fungicide, vinclozolin, I guess that's how you say it. And the pregnant mother was fed this common, you know, fungicide, and her offspring had low sperm count, poor fertility, prostate and kidney disease. So they then, the next generation didn't expose them to this fungicide at all. Were fed a normal diet. They weren't given anything additionally, but they were fed a normal diet. And the third generation, the great-grandsons of the exposed pups, had low sperm counts. So this one epigenetic change in the pregnant mother affected not only her and her offspring, but also epigenetically affected, the great-grandson of her.
Yeah, and when I first started working here, I had read about how mothers that were low in zinc would have children that were low in zinc, and even if those children ate adequate zinc in their diet, they could have children that were low in zinc. So that can be an epigenetic hereditary factor.
So, we've kind of just talked about this, the epigenetic inheritance and reprogramming. So twin studies are fascinating, and twins, are often recruited for studies because it's fascinating to look at. Here you have, two people with identical DNA, but yet, if you think about it, even though these genes or these twins have the exact same DNA, very often one of them will end up, with a chronic disease and the other one doesn't. So how do you explain that? Epigenetics. And so this is just kind of a fun graph. If you look at the whites, it doesn't show up real well on our screen, but you'll see it in your slide presentation. But here on the left, these are the white lines. And so this is, the likelihood that if one twin, let's say, height is genetically, very common for identical twins to have the same height. Well, if you look at, all of these chronic diseases, as you kind of go down to, schizophrenia, alcoholism, some of these diseases we develop later in life that are a lot based on our lifestyle choices, you can see that the likelihood of, if one twin has that, the likelihood of the other one having it is not as great for some of these other characteristics. So
each of those bars represents a set of twins, brother or sister?
I'm sorry, each of these, okay, so uh-oh.
Oh, you hit the wrong button.
I hit the wrong button. There we go. Okay. We're back. So each of these, on the left, this bar is identical twins. This bar on the right is fraternal twins. And so you can see height-wise, if you have identical twins, they're much more likely to have the same height versus, fraternal twins, which are almost kind of like brother and sister, which have about 50% the same genes. There's about a 50-50 chance that they'll have the same height. So you can see, you would think though, with identical DNA, that this would be 100%. That if one of them has, schizophrenia, the other one should have schizophrenia, if it's all based on our genetics. But again, a lot of it is based on the epigenetic changes that happen to each twin, throughout a lifetime. So
there really is a sense of hope in this, because a lot of people have come to me and said, I'm doomed because I had both parents had such and such. And in reality, that's not the case. You can make a difference in the way your body evolves based upon the choices you make, but that's what the whole field of epigenetics is about. What are the choices that you can make? Now, the book that we forgot to bring that book. Or did you bring Jack?
I didn't bring it.
Oh,
No.
There's a book called Feed Your Genes Right by Jack Chalaman. He spent quite a bit of time looking at this, and you can get into quite a bit of detail. But today, we thought we would just give you a little bit of of an introduction to some of the things you can do to improve your
Well,
genetic potential.
And to kind of piggyback on what you were just saying, to me, this provides also, you know, a lot of hope that we aren't predetermined, or you know, we have an inherited a genetic profile from our parents, but to me that even more highlights the fact that we are each biochemically individual. And for me personally, you know, talking about zinc babies, I have a very hard time, because you know both of my parents tend to be low in zinc. I have a very hard time holding on and utilizing zinc. Thanks, Dad.
Sorry, but look how beautiful she is.
So for me, you know, if I'm just taking the recommended daily allowance of zinc, I'm not getting enough. But by testing, you know, testing my nutrients, testing my individual nutrient profile, I know that I have to take about 5 times the recommended daily allowance of zinc just to maintain normal levels. So there is hope, but sometimes a little bit of guidance is needed, and you know, sometimes just taking a multivitamin isn't enough.
Right,
for
and
each of us.
One of
Individuals.
the famous doctors that spoke at an early conference when Dr. Reardon first opened the Center for the Improvement of Human Functioning, he had 15 international conferences on human functioning, and one of his famous presenters was Dr. Roger Williams, who wrote several books on nutrition. And he wrote a book called Biochemical Individuality, which means why are we all so darn different and unique in our own little way? And part of it has to do with this epigenetics. And he had a term for that back then that he called it genetotropism, which means...
Which is even worse.
Which is even worse. Epigenetics is a little easier to say. But genetotropism basically said that based upon our genetic factors, we may have enzymes that don't manifest as well. A really good illustration of this, where we can do a special test for people that have environmental disease. How many of you know, where they can't be around perfumes, or they can't be around candles or things like that, because their livers just doesn't seem to process these things very well, and it makes them sick. And people that don't have this problem don't understand it. But the people who've got it, they're pretty miserable because they have a hard time dealing with all the environmental chemicals. Well, it turns out that they have genetic polymorphisms, which are little kind of like typos in their genetic code, which changes the way in this case, their liver makes glutathione. And so glutathione is a very important molecule that detoxifies a lot of the things that we come in contact with. But they can't do it very well, and so consequently they have a much narrower realm of life that they can exist in without and still feel good. So this is an example of genetotropism. Now they can survive, though, by really amping up either the precursors for glutathione, or now we actually have glutathione as a liposomal supplement that people can take in much higher amounts. And so anyone who's got this condition, I'm having them really amp up their glutathione. So, and we can measure for that genetic polymorphism. And so the whole study of genetics, we kind of thought we would study the human genome and we'd get all these answers. Well, not really a whole lot has come out of that yet. Though now with the emerging field of epigenetics, we're beginning to see there's a lot of potential for us learning to understand how environmental factors and behavioral choices will open up this whole field of genetics for the average person.
Okay, so we've come up with 10 steps that how can you, kind of improve your odds, improve your chances of providing the right information to your genes. And so number 1 is reducing exposure to toxins. And that's kind of a broad blanket statement. How do you avoid environmental toxins? They're everywhere, from in the furniture that we sit on, to the air that we breathe, the food that we eat. They're kind of, it's rampant within our culture. So I mean, just a couple of products, I've listed a couple of cosmetic products to look out for that do tend to carry very low levels of toxins, but yet when you start adding up all of the different, sources that we have of toxins within our environment, it can add up to be significant. I've provided a website here. The Environmental Working Group has a wonderful website where you can go on and look up. They have a whole cosmetic database where they rate different cosmetics to say, what you're having, are there parabens in it, are there other toxic chemicals, and so that's a great resource that they give to kind of help guide you in what you're actually using. Replacing chemical cleaners. Really, we use all these chemical cleaners, but really, you can make your own, or buy some at the health food store that don't contain all of the toxins. The gauge I use is if there's a chemical cleaner that they warn you to keep away from children, or that call poison control if your child swallows it. Well, chances are if you're coming in contact with it, it's probably poisonous to you as well. Just you're larger than the child, so that's kind of a good gauge to use with cleaners. Reduce pesticide exposure. There's always a big question of organics. Do you do organic? Do you not? Are organics that much better than regular? And again, these are personal choices that everybody has to make. And it's kind of, I look at it. What are the things that I can control? What are the things that I can't control? Because there's exposure that you have that you can't control. But perhaps removing some of the foods that do contain pesticides out of your diet will, minimize your risk. And so I've provided, how many people have heard of the Dirty Dozen? Has anybody heard that term before? Okay, so good. A lot of people have. So the Dirty Dozen, if you were to remove these with 12, and then we've added a couple bonus ones, but if you were to remove these 12 foods, or you bought these 12 foods organic, you would reduce your pesticide exposure by about 85%. So it's a significant amount. So if you can't afford to do everything organic, these are some of the worst offenders as far as pesticide exposure. And then these are, again, the flip side to this is the Clean 15. So these are foods that tend to be pretty safe that you could buy conventional and not worry too much about. Eating a nutrient-dense diet. It's fascinating to think about the kind of the framework for our genes evolutionary-wise and how they developed. Our genes and our bodies developed to be able to work with a very high nutrient-dense diet. We tend to think of eating, green leafy vegetables and, nuts and seeds and, lean protein. We tend to think that as we're taking a proactive step in our health, when in reality, that's what our body was designed to eat. That was kind of the status quo for thousands of years, and that's what our ancestors ate. And so really, that's what our body knows how to utilize. And anything that isn't within that category, our body has to work extra hard and steal nutrients to be able to process and digest and eliminate.
Yeah, if you don't eat a nutrient-dense diet and you're stressed, you're going to be like those mice that Dr. Reardon studied, where you may not be totally without a nutrient, but you're going to dip down into some very low levels, and your body will have to compensate by calling forth epigenetic adaptation, which may or may not be good. It may be a maladaptation. I think a lot of the diseases that we have, like inflammation, very often you don't have enough omega-3 fatty acids, and so your body overreacts in terms of its inflammatory response to allergens. So the whole idea is to give your body the best you can so that you don't trigger any of these maladaptive epigenetic responses.
Drink clean water. This is a big one. There are over 300 pollutants in U.S. tap water. The U.S. government hasn't set a new standard since 2001, so a lot has probably changed. Bottled water doesn't seem to be a whole lot better, because, how many of us have had the experience of drinking out of a plastic water bottle and it kind of tastes like plastic, like we get that taste? Well, that's because, as that water is transported, it gets heated up and some of those plastics leach into the water. So that doesn't tend to be a great option either. And then, and the flip side of that too is the water bottle companies don't have to disclose where they get that water. Pure spring water could be, New York municipal water, for all we know. They don't have to disclose that information. So drinking clean water, having a good source of reverse osmosis, clean water is important. Preservatives and additives. We've provided a list here of some of the most common preservatives and additives, and I'll let you read through that on your own. But we list some of the most common ones and what types of foods you tend to find those in.
I wanted to. A lot of experts will sometimes say, but the pesticides and the additives are in such small amounts. Don't worry about it. They're too small to give you any particular problem. What the problem is what's called bioaccumulation. And one of the theories of obesity that's going on in the country right now is that we're becoming obese so that we have some place to dump all the toxins that we're being exposed to, and that may be a self-protective response. But as you eat, see, we're pretty high up on the food chain. A lot of these pesticides accumulate as you move up the food chain. So this is an example from Lake Ontario, PCBs. If you look at the phytoplankton, they have 250 times the PCBs inside them that's in the water. But the zooplankton have 500 times, and the shrimp which eat the zooplankton have 45,000 times, and the small fish that eat the shrimp have 835,000 times, and the trout that eat the small fish have 2,800,000 times. And then we eat the trout. So then it's, and the more we eat as time goes on, we bioaccumulate. I'm sorry?
Oh, I said no, we eat the small fish.
We probably, oh yeah, and the small fish is better. They say if you're going to eat fish, eat the smaller fish because there's less bioaccumulation that has occurred.
Oh, and then we have the herring.
And the herring, poor herring gull. We don't eat the gulls, but my gosh. We are the gulls. Okay. This is just I took the book Eat Right for Your Feed Your Genes Right, and looked at some of the things that you can do epigenetically to change the course of the illness. Alzheimer's has to do with these proteins developing in the brain, and now we're talking about type 3 diabetes as being a cause for Alzheimer's. Have you heard the term type 3 diabetes? Well, type 2 is where you are making insulin, but your cells have developed insulin resistance. Type 3 is where your brain, which is so dependent upon glucose for energy, it develops insulin resistance. And this is where the whole notion of coconut oil has come in, because coconut oil has the structure very similar to ketone bodies. And so when you're in ketosis, that's an alternate way to get energy to your brain. And so there's been some people that have used coconut successfully in people that have the type 3 Alzheimer's. Certainly B12 and folate, as we age, we oftentimes need more B12 and folate, and our digestive systems can't make the intrinsic factor that it needs to absorb it. So we could actually go through each one of these. I don't think we're going to have time, but each one of these, there are nutrient choices that you can make that begin to modify some of the epigenetic responses that your body has made in an abortive attempt to deal with a poor environment. Okay, go ahead. And so, number 8 here, oh, you want to go through that one?
Sure.
This is just a question, from whom do we inherit our mitochondrial DNA?
Your mitochondria is the powerhouse. It provides
The mother,
energy.
the father, or both parents? The mother? Who votes for the mother? Don't know. It is the mother, interestingly
Blame the
enough.
mother.
And a lot of times thyroid problems come down on the side of the mother, but that may be related to the DNA issue. But so anyway, take care of your mom. Choose your parents very well. So the final major, and I sometimes call it the ultimate dysregulator, is stress. And stress can take so many different forms in our lives, but yet you know it when you're in it. And you can take action to reduce it. whether it's getting more sleep or dishing out a little bit more forgiveness or being a little bit more understanding in relationships, or even just how you set up your sleep schedule. Oftentimes, we are the major cause of our stress. And so what we know is that when people are under extra stress, they use up nutrient reserves faster, and it does call upon these epigenetic programs more as the body attempts to adapt to the environment. And you could really say that epigenetics is the human or any living organism attempting to adapt to its environment. And if it has the resources it needs to make the adaptation, everything goes fine. But if you don't have those resources, if you don't have what Dr. Reardon called the reserve to do what you need to do and want to do with energy and enthusiasm, you tend to get sick. And I think really many, if not most, of the modern illnesses are diseases of maladaptation because of environmental challenges and a lack of adequate nutrients and resources in order to meet the challenge of those situations.